So before moving into large insert vectors, let's have a brief recap of what we had done in the previous lecture. So in the last lecture on vectors, we discussed Lambda Phage and M13 vectors. So this is a Lambda Phage, which is typically a rocket-shaped virus which can infect the bacteria. The genome here is linear in the virus and becomes circular in the bacteria by the hybridization of the complementary cos sites of single-stranded nucleotide extensions present at ends on either strands of the DNA here, right? So these are our cos sites. What is also important is that the genes are lysogenic life cycle are found here and can be completely removed, and this region can be replaced by your DNA of interest. So depending on whether a part of the lambda genome is removed or not, you have two types of vectors that are available with respect to lambda Phage. One that is known as the insertion vector and the other that is known as a replacement vector. In the insertion vector, the basic lambda genome is maintained and the insert or the DNA of interest is basically pasted in a region within the C1 gene. Because the C1 gene gets disrupted, the virus cannot undergo the lysogenic life cycle and only maintains the lytic life cycle, amplifying your piece of DNA. The other type of vector is what is known as the replacement vector, where a part of the lambda Phage DNA is completely removed. And because a part has been removed, a larger fragment of DNA can be inserted here in this region. So this is the replacement vector. In the insertion vector, the largest insert size is around 10 kb, not more than 10 kb of insert can be successfully cloned in an insertion vector. In comparison, in a replacement vector, you could have a fragment as large as 22 to 23 kb that can be successfully cloned into the replacement vector, right? So this is your lambda Phage, and then of course we talk more about another important vector that is known as the M13 vector. M13 vector is important because here the DNA is circular and very importantly the DNA is single-stranded. In comparison, the lambda Phage DNA was double-stranded. This is helpful in generating single-stranded DNA fragments and therefore is of significance where you wanted to have a single-stranded DNA for sequencing and also for development of probes, right? This is the M13 genome here, around 6407 bases. And if you look into this, there is an M13 origin of replication and there are 10 genes and all genes are essential for the survival of the M13 vector. So therefore, there is nothing that is dispensable here, as was the case in case of lambda Phage, right? And if you look into the evolution of M13 vectors, origin of replication is found in between gene 4 and gene 2. And initially, a LAGZ gene was introduced here for blue-white screening. Within this LAGZ gene, an eco-R1 site was introduced. And later on, a multiple cloning site was introduced within the LAGZ prime region so as to ensure choice of restriction sites that can be used for directional cloning. Towards the end of the lecture, we talked about PhageMed, which is basically, as the name indicates, a combination of Phage and plasmid. And here, if you see, you have a Puck origin of replication and you also have an F1 origin of replication. Then towards the end, we discussed COSMID. COSMID, as the name indicates, is a combination of the cos sites and the plasmid. So here you are, this is your cos sites, this is your resistance marker, this is the origin of replication. And then you have your additional restriction sites. And the advantage with COSMIDs is that because it has cos sites, it can be packaged in vitro. And because cos sites require a certain threshold length of DNA to be present, you can actually package around 50 kb of DNA into this plasmid here. So this is where we were in the last lecture. Now we move on and we talk about large insert vectors. Now the first question is, why would you want to have a vector which can hold more than 50 kb of DNA sequence?